Hello, students. Welcome to Chapter 16. Chapter 16 is about how populations evolve over time. And we're going to take a look at three sections: genes, population, and evolution, natural selection, and maintaining diversity within the population. So let's get started. The first thing to do is to take a look at your knowledge of these objectives, explaining evolution in populations and allele frequency. What are the 5 conditions for Hardy-Weinberg? Can you apply what you now know about that Hardy-Weinberg equilibrium? And then what are the agents of change that cause evolution? And then after this section, come back to these objectives and then rescore your knowledge. And that will give you a good idea of what you want to go back and review or ask questions about. So, a population is just a group of organisms of one kind of species in a certain place at a certain time. And it's the scientist who is studying this population that will determine that place and that time. And then that scientist may be a population geneticist studying population genetics, or the diversity of the gene pool of that population. And we look at that as the amount or different percentages of alleles that are in that population. And it will give us the diversity of the population because our genetic code, our genotype, will then determine the physical characteristics of those living things. And we can see how that gene pool and those physical characteristics change within the population over time. And when we are studying that change over time, we're really studying microevolution. So, microevolution is just the change in the gene pool of a population. And what we know is that there's already an inherent diversity within a population. So, there's various alleles at these different gene locuses, and that is what gives us our gene pool. I like to think of a population sort of like a deck of cards, and every new generation is just reshuffling that deck of cards. Um, and so we can describe our population as that genotype frequency and then the alleles. If we remember, genotype, that's your genetic code. Are you homozygous dominant? Are you heterozygous? Are you homozygous recessive? What is that actual genetic code? And then your alleles are dominant alleles and recessive alleles. So here is another way to sort of look at that idea. If we're looking at, say, peppered moths, we know that there is a physical appearance to these moths. They either have a dark color or they have this light speckledy color. And what we see is that the genotype, the genetic code, if you are two dominant alleles, hetero or homozygous dominant, then you'll have that dark color. If you are heterozygous, one dominant allele, one recessive allele, you also show that dark color. And then if you are homozygous recessive, then you show that light speckled pattern. And when we're looking at the population, we can look at those genotypes. How many individuals are homozygous dominant, are heterozygous, or are homozygous recessive? And we can look at the alleles. How many dominant alleles are there? How many recessive alleles are there? So let's start with that allele frequency. Frequency is just another way that we can describe percentage or amount of. And so it is the proportion of each allele. How many dominant alleles, how many recessive alleles. And we need that number to equal the entire population. So we say that the amount of dominant alleles, the amount of recessive alleles must equal 1 or 100% of the population. And we can actually write a mathematical equation to express this. So we'll say P plus Q. P is the percentage or frequency of dominant alleles. Q is the percentage or frequency of recessive alleles. And that is 100% of the population. And then microevolution is just calculating a change in these allele frequencies over time. So how this would work in the real ecological world is ecologist would go out, sample a population, and figure out how many dominant alleles P and how many recessive alleles Q are in that population. And then they would leave that population alone. Then they could come back at a later time, calculate again that P value or the dominant allele frequency and that Q value, the recessive allele frequency. And if those numbers have changed, then it means our population has undergone microevolution. Now we're no longer just shuffling the deck of cards, but we've inserted new cards into that deck. If the population frequencies or the gene frequencies do not change when that scientist came back and recalculated, then no evolution has occurred. So here is exactly what that would look like. So you are an ecologist, you've gone out and you've counted the population and you've determined from that that our dominant allele is there 10 times, our recessive allele is there 40 times, and then you do some math and you find that your p-value is going to be 0.2 because that's 10 out of the 50 total that you counted, and that our Q value is 40 out of 50, and that is 80. So when we add 0.2 and 0.8, we get 1. There is going to be another equation we will see in just a moment. This is my p squared plus 2pq plus q squared equals 1. And that now is not just dominant and recessive alleles, but that's actually my genotype frequency, where p squared is how many homozygous dominant individuals are there. My 2pq are how many heterozygous individuals there are. And my q squared is how many homozygous recessive individuals there are. Now, for my course, my class, I do not require you to calculate Hardy-Weinberg equilibrium in lecture or lab, but be sure to see your instructor, if it's not me and you're watching this video, so that you know if you have to do the math. So in order for a population to be studied with Hardy-Weinberg equilibrium, that's those math equations, we must assume that our population is constant, that it's not changing. That way, when we come back and calculate that allele frequency again, if there is a change, we can say, okay, the population is undergoing evolution. So what are the conditions that must be met for Hardy-Weinberg equilibrium? Well, the first is that there are no mutations. If there is a mutation, that would change the gene pool and that would cause evolution to be occurring. So there can be no mutations. There can be no gene flow or no migration in or out of the population. If individuals are entering or leaving, that would change the gene pool. So we cannot have that. There needs to be a large gene pool or there can be no genetic drift. We cannot have a small population because when we lose a bunch of individuals, well, then we've changed the gene pool. There must be random mating, or in other words, uh, there's no non random mating. You don't choose your mate, it's just a random chance. If you chose your mate, then you would be selecting certain genes to be better than others, and we cannot have that. And then there's no natural selection when we assume Hardy-Weinberg equilibrium. If nature is choosing better genotypes and therefore uh, or phenotypes and therefore genotypes, there would be change, and we cannot have that. Now, as you look at this list of no mutation, no migration, large gene pool, random mating, and no natural selection, you're probably thinking, this isn't happening in a real population in the wild. And for the most part, you are correct. This is a hypothetical situation that we are saying is true when we calculate Hardy-Weinberg equilibrium, so that when we come back and get a new calculation, we can say one of those things or more than one of those things is happening. And this is a very important idea for supporting evolution because we are seeing and calculating with math that the population is changing. So it gives a lot of support and a lot of evidence to the theory of evolution by natural selection. So here is sort of the idea of how we would calculate what we would expect to see. Again, my class, you are not calculating. If you are watching this video from a separate class, be sure to talk to your instructor. But what we would see is you have this 0.2 or 20% chance of passing on this dominant allele, 80% chance of passing on this recessive allele. What happens when we cross those? We're finding that only 4% or 0.04 would then have that homozygous dominant characteristic or genotype and give you that dark characteristic. We would find 16 plus 16, so 32 percent of the population would be heterozygous. They would look that dark color, but they would have that hidden recessive gene. And then we've got our homozygous recessive, where we have our speckled light colored, and that's 64 or 0.64 in the population. So again, in order to understand Hardy-Weinberg equilibrium, we need to remember that those conditions, the no mutations, the no gene flow or migration, the random mating, the large population size, or no genetic drift, the no selection, that is rarely, if ever, met. And so then we can say any deviation from Hardy-Weinberg equilibrium means that evolution is occurring. We're analyzing the allele changes in the population over time and showing mathematically that evolution is occurring. So, for example, we may have these moths, and we see that they are those dark colored moths most of the time in the population. They're being eaten. And so that would be natural selection, right? They're not able to have the best characteristics for their habitat. Their genes make them be a color that is easily spotted. They're eaten. And now, when we come back to the population and do our calculations again, we'll see that those numbers, those p's and q's and p squared and 2pq and q squared, they've changed. And therefore, our Hardy-Weinberg equilibrium was not met. Those conditions were not there. Evolution has occurred. So, how do we determine if the evolution has occurred? Well, if we look at each condition, such as Hardy-Weinberg says that there is random mating. If there is non-random mating, if individuals are choosing their mate, then those alleles are not random. And so then we are seeing a change in what alleles are in our gene pool, and we would expect there to be a change in our calculations. If there is no natural selection, the deviation is that there is selection, and certain alleles will show up more. It will change our allele frequency, and we would calculate that yes, there's a change to Hardy-Weinberg equilibrium and evolution has occurred. There's no mutations is the condition. Well, the other thing that could happen is there are mutations. We would get new alleles that changes our allele frequency, and yes, evolution has occurred. Migration cannot happen with Hardy-Weinberg equilibrium. If there's immigration come into the population or emigration leave the population, those individuals take with them their genes. We would expect a change in our allele frequencies, and yes, evolution would occur. If we have a large population, or we say there's no genetic drift, that's the condition for Hardy-Weinberg. But the opposite of that, or the deviation, is there is genetic drift. We have a smaller population size, and we're losing individuals. Alleles may even disappear, and then that would cause evolution. So, how do we get those deviations, or what are they? So, I like to use my hand and think of my fingers when I think of a way to remember these mechanisms that cause evolution, or are the um, reasons behind a change in that gene pool. And the first one is mutation. And I like to think of my middle finger, M for middle, M for mutation. And a mutation is just a change in your DNA. And that change within your DNA can provide new alleles. And some mutations um, are bad, and they won't stay in a population. Some don't do anything and they may or may not remain, but some are more adaptive, and then we will see more of that in the population over time. So, for example, genetic mutations that affect the pigment in the peppered moth wings gave variation, and then we saw if you are able to hide better on the bark because you're a speckledy moth, well, then you're more likely to survive. So, again, I think M for middle finger, M for mutation. Then I think of gene flow or migration, immigration, emigration. I like to use gene flow, and I use my pointer finger because my pointer finger can point in and out and move between my populations. So, gene flow is just the movement of alleles within that population when we're carried into or out of the population. And one place that we see this quite often is plants. When they're carried, the seeds or the pollen is carried into a new population. It is a big concern when we're looking at genetically modified plants that are being farmed and we're putting them right next to um, more conventionally grown plants that aren't genetically modified. And what if that pollen, what if those seeds can move into those plants that are not genetically modified? Would that change both species? Is that, something that's happening? What we see with gene flow, again, my pointer finger, is that it can have individuals moving back and forth between populations. And when we move those individuals back and forth, we don't see the gene pool separate. They're still in the same gene pool. If that migration, that gene flow doesn't happen, then our populations become more isolated over time. Their gene pools become different, and then they're isolated and they won't reproduce with each other. And really, that's the first step in getting a brand new species is isolating these populations. And when we put them back together, they won't mate with each other any longer. So here's our example of some gene flow. Maybe we've got our peppered moths and they're in an industrialized area. So it's actually better to have those dark wings so you can hide in sooty smoke. And then if you're a little peppered moth in the countryside, it's better for you to have the speckledy peppered wings because you don't have any smoke to hide in, and the bark of the trees is light. But if those moths can interact with each other, we're having that gene flow occur. We're keeping that gene pool diverse, and we're not isolating, we're not creating a mechanism for evolution. Then genetic drift is this extreme population size change. I like to think of my pinky finger for genetic drift because my pinky finger is small. So when I see genetic drift, it is a change in the allele frequencies because there is something that has occurred to drastically shrink the population. And this doesn't necessarily lead to adaptation to the environment, but what happens is we had a large, diverse gene pool, and then we can eliminate some of those alleles from the population. Or we could see alleles that were kind of uncommon, but now they're much more common because we shrank that population size. There's sort of two ways that we see this happen. There is what is called the bottleneck effect. And that is where, we have this large population and then something happens where only a few individuals, like a few that you could pour out of a glass bottle, a few of those individuals survive. And their gene population or their gene frequency in their population is very different than that whole bottle of marbles or rocks or whatever it was that you had in there and you poured just a couple out. um, then we may get some severe inbreeding, because we've really limited our gene pool. We also have something called the founder effect, which is where we have a few individuals that will establish a new group, a new population. And then when that happens, only the genes that were brought there survive. And what we see is we, again, could lose or fix some allele combinations. So here's a great example. We've got our little moths, and they're in a forest, and then there is this forest fire, this big, traumatic event that drastically shrinks the population size. And what we see is, we had a lot of light colored moths and just a little bit of this other darker colored moth. But then after the forest fire, when a lot of those light colored moths died, now the percent of our dark colored moths within the population is much higher than where we started. And this is that bottleneck effect. It is where we have a large population, something happens, we lose some great number of our population, and it changes the allele frequency. And it's usually some kind of random event, a fire, a flood, a landslide, something that then prevents the majority of individuals from surviving, getting to that next generation. And then the alleles are different. Again, founder effect works similarly, but is not this big natural disaster. It's just that a new population is started from a few individuals, and whatever individuals are those founders, well, their genes are the ones in the new population. A great example of this is if we take a look at Amish communities in the United States. They tend to stay within their communities and not, leave and bring in new genetic code. But when we have, that founding here in the United States, a group of people from Europe came with their genetic code that was different than the population we saw in Europe. They come to the United States, they found an Amish community, and now we see that there are certain genetic diseases that are more common just because that was the gene pool by random chance that founded this new population. And so, again, the greater the reduction in the population size, the bigger the chance we will change the allele frequency. It could be at a higher frequency, such as Amish community having certain types of dwarfism, for example, or we could just lose alleles from the population. Something else that can occur when we have this founder effect, if you think of it in the sort of animal kingdom, you have, a population of penguins, and then the iceberg they're on breaks, and a little group of the penguins, drift away. That can have a founder effect. That's a new population, but they'll be higher than normal occurrences of inbreeding. And then we are even more likely to see rare genetic disorders occur because we have such a small gene pool. And this is happening to many species where their habitats are being destroyed, things like cheetahs, penguins. We're seeing this, drastic change in population size and this inbreeding and more genetic disorders. So again, here's our picture to show us this bottleneck effect. So you have, this bottle of marbles, you pour some of it out, you lose alleles, you change the frequency in this new population. All right, so to recap and recall, right, we've already had mutations, that's our middle finger. We've already had our gene flow or migration, which is our pointer finger. We've had our genetic drift or founder effect and bottleneck effect, that was our pinky finger. Let's look at our ring finger for non-random mating, right? Think about someone getting engaged, getting married, they put a ring on that ring finger. So when individuals do not choose their mate, it's random, right? And the gene pool just shuffles like a deck of cards. However, when individuals are choosing their mates, then it's non random, right? When you are picking your partner, you are looking for certain kinds of traits. Sometimes we can have assortive mating where individuals try to select mates with the same phenotype. They'll reject differing phenotypes. And this can be important. There's an example in your textbook of non random mating increasing color blindness in individuals. So, this is still a type of non random. You're still choosing your mate. And then we are going to have an entire section dedicated now to that last mechanism, which is your thumb, which would be natural selection. Nature chooses thumb up or thumbs down for your um, traits to be good for the environment you're in or not so good for the environment that you are in, which is this section right here. We're going to take a look at stabilizing, directional, and disruptive selection and determining natural selection. So let's go ahead and do that. So natural selection is the adaptation of a population to the environment, the living parts or biotic parts and the non-living parts, abiotic parts. In order for natural selection to occur, you must have variation already within the population, that the individuals in that population already have differences. Those differences must be able to be passed on through the genetic code. And then those differences must affect your survivability. If you have characteristics that allow you to get more food and shelter and space and water, find a mate, well, then you are more likely to reproduce. Whereas if you do not have those traits that allow you to find more food, water, shelter, space, and a mate, you are less likely to survive and find a mate. And then, of course, those differences will affect how successful you are at reproducing. Natural selection is going to favor the genetic code, the variant that is most adaptive to the present environmental conditions. But we know those environmental conditions can change, and then a new trait might be better. And then natural selection will work to choose that trait. So we really see three kinds of natural selection happening. We see what is called stabilizing selection, where we may have two extreme physical appearances. Maybe it is a really tall, long stem of a plant. Then the other really far extreme is a really super short stem of a plant. And then right in the middle, stabilizing, is that medium sized stem of a plant. Then we can have directional selection, which is where we're pushing a certain kind of physical appearance. Maybe there is our dark colored moths and our light colored moths, and normally, there's an amount of each, but now I'm pushing and seeing more of one of those physical appearances. And then the least common is our disruptive selection. That's where I have my normal, variability. But now I'm choosing the two extremes. If we think back to the original example I gave you with a short plant and a tall plant and a medium plant, instead of choosing the medium stemmed plant, nature's choosing tall and short. So let's get into these. So, what we know is that your genetic code is controlling that physical appearance. And most traits are polygenic, or that there's more than one gene at play to give you that physical appearance. And so we get this sort of bell shaped curve, right? For the most part, we're not looking at, for example, only red flowers and only white flowers. There's red and pink and white. That would be the bell curve kind of thing, which then can lead us to something called stabilizing selection. That's where that intermediate, that middle phenotype is the best for the environmental conditions. And so we see the peaks, the other parts of that variation kind of shrink away to stabilize us at that middle. A great example is human babies. So when babies are born, they're all about, 7 pounds. Babies historically that were born that were like 3 or 4 pounds, they did not survive. Babies that were 10, 11, 12 pounds historically, they did not survive. But if you were a 7 pound baby, you were big enough to survive in the world, easy enough to be birthed. That was that stabilizing selection. Most babies were born about 7 pounds. That's what we see here. So if you're really small, don't survive. If you're really big, also don't survive. But right in the middle, there is that stabilizing selection. Now, with our NICUs and C-sections, we've kind of messed this up. And we can have babies that are 3 or 4 pounds survive. We can have babies that are 10, 12, 13 pounds survive. But if you are naturally birthing a baby, that's still about 7 pound range is going to be that stabilized selection, that best, you know, for the environment situation. Then there's directional selection, where we're picking a phenotype, pushing the curve in that direction. A great example is bacteria that are able to be resistant to antibiotics. We just need one to survive that course of antibiotics. It can reproduce, and now all of that bacteria that came from that one that was able to survive, it now is all able to survive the antibiotic course. So we've pushed our selection towards that phenotype. Then there is disruptive selection, again, where the two extremes are what is favored. The curve gets sort of this bimodal, two peaks. And really, the example from your book is the only one that I've ever heard of. So this is pretty rare. There are land snails that live in forest and live in sort of flat grassland in Britain. And if you are a snail that lives in the forest and you have a dark coat or a dark shell, then you're able to hide in the leaf litter. If you are a land snail and you are in that grassland and you have a light colored shell, you're able to survive and hide in the grass. But if you have that middle color that's like kind of stripy, not dark, not light, you are eaten in the forest, you are eaten in the grassland, and you are not selected for. So here's our example of our bacteria. So we've got a couple drug resistant bacteria. Over time, those survive the antibiotic, and now we've pushed our population toward that antibiotic resistance, that genetic code. Here's my land snails. If you've got a dark shell, you can hide in the leaves. If you've got a light shell, you can hide in the grass. If you've got this middle colored shell, you cannot hide in the leaves. You cannot hide in the grass. You are eaten. So there's a lot less of you, but there's more of our two extreme color patterns. And then a type of selection is actually called sexual selection. It's where males and females have changes or adaptations that can increase their ability to find a mate. With males, there is this increased ability to compete. Females are going to select or have female choice for that fitness. Who is the best able to survive and produce offspring? So, female choice is this undertaking that females are given because they produce very few eggs, right? When we learned about meiosis, we saw that for every, one cell that starts the process of meiosis, we get one egg. But for males, for every one cell that starts the process of meiosis, we get 4 sperm. So we get to be choosier with that only having one egg. We want to make sure it's the best possible mate that we can find. So there's what's called the good gene hypothesis, where females choose the mate based on traits that improve survival. There's also called a runaway hypothesis, where females choose mates based on traits that improve the male appearance. Either way, it's this female of that species that is choosing which male they will mate with. There is also sexual dimorphism, that males and females look different, whether that's in size or other traits. It explains why the male birds are more ornate, and they will, have these pretty feathers and do intense dance moves to try to attract the female and make her choose them. So here's that example of males and females looking different. That's that dimorphism. And then, of course, there is the male competition. Males can father many offspring because they are, again, making more sperm, 4 sperm for every one cell that starts meiosis. So they are really competing in order to find females to inseminate. And there is a cost benefit analysis that they have to go through. Is access to mating worth the competition? And for younger males of a species, they may have to sort of wait within the population or try to establish their own population. But there will be some sort of competition that occurs in order for the females to then make their choice. All right. And then our last section here is can we get some examples of maintaining that diversity within a population? Now that we know that, we can mathematically calculate this with Hardy-Weinberg, what are these conditions that we need to meet, or mechanisms that explain how we get to evolution? How do we maintain diversity in our populations? So, the thing about a population is that the variations are limited. It's whatever our genetic code is. And we can get mutations and we can get new individuals. Our population size may change, but ultimately, we are limited because those changes could be useful and lead to adaptations. They could do nothing or even be harmful. So it is an advantage to maintain diversity, to keep that variation and not push all of our population towards one phenotype or genotype. And so we want to see how that is possible. So the first thing to remember is that natural selection is going to be this idea that your genetic code determines your physical appearance. And then the environment is going to thumbs up or thumbs down those traits. And if it's thumbs up, well, you get more resources, you are able to reproduce, and then your genetic code is passed on. And if you do not get that thumbs up from the environment, well, then you don't get enough resources, you don't reproduce, that genetic code is not passed on. But evolution can't just start from scratch. Again, we need to have these changes in the genetic code, this variation for natural selection to work. So it's kind of an imperfect system. There's no real direction towards perfection. It's just what can the population have right now? What makes it best to survive? A great example is, humans, we are primates, but we stand upright. We're bipedal. And we use our hands instead of like a limb like other primates do. We use it to hold stuff and carry stuff and, do tool usage. Um, but when we freed our hands to do that, from going from a four-limbed thing to two limbs on the ground, that causes a lot of spinal injuries. Um, and if you are a young person, enjoy it, because every day of my 30s has been back pain, because that erect posture puts more pressure onto your lower back, and causes pinching. Whereas if we were a four-limbed creature, like other primates, maybe we wouldn't have that. And that's an example of this imperfection of natural selection. It was best to have our hands free to use tools and have fire and to carry stuff, but it came at a cost. And so disruptive selection, because of in the environment, actually gives us these sort of multiple forms in our population. And if a population is in a very big range, we can even get little subpopulations, and we may even call them subspecies. And so the environments can change, and then maybe those characteristics in these subspecies, well, now that is what is a better adaptive trait, and we may see more of. So the environment includes these selecting factors that, really do keep that diversity. A great example of how this works, because, just knowing the words is okay, but I think the understanding what we mean is more important. We've got Galapagos finches. And we can have an average beak size when we put a bunch of those finches on an island together with food. But then we see if we separate them on different islands, well, then the average beak size varies. So, I guess let's first take a look at this subspecies thing. We've got rat snakes, and we've got many different kinds. We've got black rat snakes, yellow rat snakes, gray rat snakes, red rat snakes, also called corn snakes. And what we see is they really overlap in where they can live. And it may be better here in Illinois to have a black rat snake with its coloration. It can hide in the dirt, it can climb up the trees, it finds its food. But then here in South Carolina, we more so find yellow rat snakes. And if we think about our palm trees and our sandy soil, well, it makes sense that a black rat snake might not be the best here. We've got a kind of lighter colored soil. That being said, their populations do overlap. We do see black rat snakes still here in South Carolina. Um, and they're just not as common because we have yellow rat snakes. Now, if we put a yellow rat snake and a black rat snake together, could they mate? It's very possible. Um, and then we could get something else. But we're keeping this diversity within our gene pool because we have both subspecies, even though they really are not a different species at this point. They can still interbreed with one another. They just maybe won't if they don't interact with each other and find each other. We also see that we can keep recessive alleles within a population when we have our heterozygotes, right? They hide or shelter that recessive allele because the dominant allele is the phenotype that we see. So even alleles that, when together, those recessive alleles could be lethal, they'll stay in the population because the dominant allele will hide it. There is something called the heterozygous advantage, which is where sometimes recessive alleles are better to have hidden by that dominant allele. The most famous example, and one that affects us here in South Carolina, one that you'll see in lab, is sickle cell disease. Sickle cell disease, or sickle cell anemia, is caused when you get two recessive alleles for sickle cell trait. If you get two recessive alleles and you get sickle cell anemia, your red blood cells are shaped like little moons or little sickles. They get clogged up in your arteries and veins. You do not get enough oxygen to your tissue. It's not good. If you get two normal non-sickle traits, then you are going to have normal red blood cells. They look like round little O shapes. However, what we see is that malaria, which is spread by mosquitoes, it's a protist, it lives inside of your red blood cells. And if you have no sickle cell trait, then you can get malaria into your system, into your red blood cells, and that's really dangerous. You can die of malaria. If you get one sickle cell trait and one normal red blood cell trait, you're a heterozygote, and you are more likely to not get malaria, and you have enough normal red blood cells that your sickle cell blood doesn't, clog your arteries, doesn't give you the symptoms of sickle cell anemia. So in that way, being a heterozygote is the best. It's an advantage because you will not get malaria, but you will not have sickle cell anemia. Where if you have two sickle cell traits, you get sickle cell anemia. If you have no sickle cell traits, you get malaria. So what we see is that there is a huge overlap of where sickle cell trait is found and where malaria is common. A very interesting historical thing is that we also have a high instance of sickle cell trait in South Carolina. and really in the south of the United States as a whole. And if you think historically why that might be, well, we took people who were living in these areas where sickle cell and malaria overlapped and brought them to the United States. And therefore, we really did a founder effect kind of thing where now sickle cell is very common, despite us not really having malaria as prevalently as we did here in Africa and parts of Asia. All right. Well, thank you so much for watching. I hope that you have enjoyed our presentation. As always, if you have questions for me or for your instructors, please reach out. Otherwise, happy studying.